Welcome to Twist Bioscience Application Support video series. Today in this video, we will be highlighting the Twist RNA sequencing solutions. To start with, why we sequence RNA, why RNA sequencing is important in some aspect of biological process. So the most important is the functional studies. As you know, genome may be constant, but an experimental condition can have a pronounced effect on gene expression. For example, wild type versus knockout mice. Secondary, RNA-seq is very important for predicting transcript sequence from genome sequence, which is difficult. Especially gene annotation is revolutionized by RNA sequencing. At the last but not least, some molecular features can only be observed at the RNA level. For example, alternative isoforms, fusion transcripts, RNA editing. So RNA sequencing has already been trendy in multiple applications, which start from oncology and until now is expanded to RNA therapy research. There are multiple aspects of RNA sequencing which is currently used not only in oncology field, but also in infectious diseases research, for example, creating antibodies for SARS-CoV-2 and also in microbiology research. So in nutshell, RNA sequencing is a powerful tool to understand the transcriptome. Twist RNA workflow expand a vast majority of the products which start from whole transcriptome RNA sequencing, which focuses on complete coverage across transcripts. It also utilizes or detect splicing or isoforms. There can be multiple applications of whole transcriptome RNA sequencing, deep transcriptome profiling, non-coding RNA profiling, analysis of coding and non-coding readings. Twist RNA workflow not only provide whole transcriptome solutions for eukaryotes, but also transcriptome target enrichment, which focuses on capturing and enriching entire transcriptome. Along with that, you don't need a lot of sequencing read, but you get more data with the fewer reads. Here, the application can be detection of low expressed genes, enrichment of coding regions for all unique sequences. Along with that, especially in the field of oncology, you can utilize transcriptome target enrichment for fusion detection. As you all know, Twist is synthesizing DNA, so we are not only limited to the fixed panel like RNA transcriptome target enrichment, but also we can create focused targeted RNA-seq as per your requirement for your research project or diagnostic need. Focused targeted RNA-seq is very popular when you want to sequence fewer reads and more sample pooling. And also it's very popular when you want to sequence genes or fusions for target of your interest, especially in field of oncology. Twist RNA sequencing workflow solution. So as I mentioned previously, that Twist Bioscience provide whole transcriptome solution, targeted RNA sequencing solution, which is rely on the fast and easy RNA library prep reagents, flexible workflows, and the modular kits. It can also work with wide range of input and RNA quality, very automation friendly. We already have scripts for different automation platform for RNA library prep. Also, we have working on developing twist-targeted enrichment optimized with exon-aware design. Twist RNA library prep kit is quite fast and efficient. It starts with rRNA depletion, which is very crucial if you want to deplete the eukaryotic rRNA and globin RNA. After the rRNA depletion, it leads to fragmentation based on heat. And once the RNA fragmentation is done, they involve first strand synthesis, which require reverse transcriptase, which is RNA-dependent DNA polymerase, which makes from RNA to the single-stranded cDNA. Then utilizing DNA polymerase, we create double-stranded cDNA. Once double-stranded cDNA is ready, you start with N repair followed by detailing, which is again one-step reaction. Once you have detailed cDNA fragments, there is adapter ligation, and once the adapter ligation is done, you start with library amplification. This entire workflow is very fast, single-day library prep workflow. It also required fewer SPRI bead purifications. And as I mentioned during the workflow description, it combines second strand synthesis and A-tailing in one step. It is also automation friendly. Whole transcriptome replicates across different runs. You can appreciate when we tested this twist RNA library workflow with 1 nanogram of RNA and also 1 nanogram of degraded RNA, the number of genes was really similar, which shows that efficient transcript detection even at low mass. It has high concordant expression across different range of input. As you can see, we have tested from 1 nanogram to 100 nanogram, and R square value is morely very close to 1. Also, as we are working always on developing a new product or improving current product, our R&D team have developed exon-aware design for RNA, which means that you can utilize our exon-aware design to detect novel fusions, which is limited when you are doing amplicon-based RNA fusion detection. So Twist RNA exome is exome built specifically for human transcriptome. You can obtain differential expression and gene fusion discovery together. Additionally, it increased sensitivity, especially for your precious FFP samples. And most important, it saves sequencing costs up to 1.5x compared to whole transcriptome profiling. As you can see in down figure, in our standard DNA panel, we have a standard panel design where the novel fusion can be dropped because of our design. However, in our exon-aware design, we densely tiled entire genes so that there's any new variant or the fusion that can be detected. The advantage of this exome enrichment that you enrich relevant transcript reads with the less total reads. In the first bar graph, you can see you detect more genes with less reads. For example, for 10 million reads for whole transcriptome, you can already get it with the twist exome enrichment 6 million reads. So with 6 million reads, you can achieve that coverage with the whole transcriptome that you get with the 10 million reads. In the second figure, you can see that it really focuses on exons. So when you use twist RNA exome, you have 99% of CDS region, which is not possible with whole transcriptome sequencing and also with 3' counting method. As you know, the on-target rates are very important in the NGS target experiments. So compared to competitor 1 RNA exome, you can highly appreciate that with our twist RNA exome, you reach percentage unique on-target rate as high as 80%. We also compared RNA exome to poly-A sequencing. As you all know, both RNA exome and poly-A RNA sequencing work by isolating coding transcript. RNA exome is considerably more efficient than poly-A in terms of recovering coding sequence, as you can see in the bar graph, where RNA exome end up in being 85% of coding regions and poly-A only end up with 25% of coding regions, which shows that RNA exome cover 87% of bases in RNA exome compared to 30% of bases in poly-A sequencing. Also, RNA exome is able to enrich for the majority of all transcripts, while poly-A sequencing target primarily the 3' UTR. In the second figure, you can see that poly-A sequencing is biased towards the end of the 3' end of the transcript where the poly-A is there. Hence, you don't get uniform coverage. In contrast to poly-A, twist RNA exome end up in uniform coverage. So the bottom line is RNA exome is more efficient and captures more relevant content, especially in context of calling variant from RNA. As I mentioned in my starting slide that we are also focused or Twist Bioscience can specialize in DNA synthesis, and we are also working with the key service provider in field of genomics. Along with the CGET, which is a key diagnostic company in Germany, we created a Twist Alliance CGET RNA fusion panel, especially for oncology. The panel features target detection of known events and also novel fusion partners. It covers 160 known fusion genes. Along with that, design includes fusion from greater than 30 different cancer, and also to target therapy cancer research, this content also include more than 80 therapeutically actionable fusions. Fusion events are associated with number of cancer types, and many of them are considered key biomarkers for precision medicine. Which method is better? Hybrid capture is always a better option for fusion detection. In this slide, you can see that there are three key metrics which makes hybrid capture a better option compared to amplicon method. First is flexibility. Hybrid capture is easy to scale and customize additional fusion targets. In contrast, amplicon is extensive optimization required for additional target and also is time consuming for testing. Second is performance. Performance is for hybrid capture is really good with the uniformly captured all the target, very important when you are working with cell-free DNA or cell-free RNA, with FFPE RNA. In amplicon method, you can see allelic dropout, and also there's sometimes mispriming if the sample is degraded. Last but not least, the discovery. Hybrid capture method can be used for novel gene discovery. However, with amplicon, it is not adequate for novel gene discovery. Again, a short recap. Twist RNA sequencing workflow solution is fast and easy method to understand the gene level or also transcriptome level of RNA expression. It can be used either as a whole transcriptome with rRNA and globin depletion included, along with RNA library prep kit. It's very flexible and also modular kit. It can work with wide range of input and RNA quality. As I already mentioned, that Twist RNA sequencing solution works from 1 nanogram until 100 nanogram of RNA. It's independent of the RNA quality. It is automation friendly. And also we can do twist target enrichment optimized with exon-aware design. In that case, we can create a twist custom panels, especially in field of oncology for fusion detection. As well as we believe in collaboration. So we also have Twist Alliance fusion panel together with CGET. Thank you.